hear me. If there's a problem hearing or any technical difficulties, please send a note in the chat box and we will try to address those. So today I'm going to be talking about adaptive clinical trial design and trying to lay out general principles in the design of a clinical trial that learns from the experience that occurs during the conduct of the trial in order to make the trial more efficient, more ethical, or more likely to give us the correct answer when we're done. I have a number of important financial disclosures. The most important of these is I work as the senior medical scientist at Berry Consultants. I'm compensated and my institution is compensated for that work, and that group works specifically on adaptive clinical trial design. In addition, I get support from a number of other groups, all related to adaptive clinical trial design or trial design in general. When you're designing a clinical trial, there's a lot of uncertainty regarding how best to treat patients within the trial and how best to measure the benefit or the effects of that treatment. And that uncertainty creates a lot of uncertainty with respect to the clinical trial design itself. And yet, ironically, we traditionally assume that all trial parameters need to be selected at the beginning when we know almost nothing about the patients who are going to be enrolled, and then held constant during the execution of the trial. And this leads to an increased risk of a negative or a failed trial, even if the treatment is inherently effective and has a clinically important benefit in at least a subset of that population that we're interested in. And when I use the term a failed trial, I mean a trial that at the very end leaves you still wondering what the correct answer is to the question regarding the efficacy of the treatment. However, once patients are enrolled in the trial that you're conducting and their outcomes start to be known, and their outcomes may be the primary outcome of the trial or the safety outcomes, that accumulating information reduces our uncertainty regarding the trial parameters that we didn't know much about when we tried to design the trial. An adaptive clinical trial is a trial that is designed prospectively to take advantage of that accumulating information during the conduct of the trial and use that information to modify trial characteristics, so in response to that accumulating information according to predefined rules. We want to do that in order to make the trial more efficient or more ethical, or perhaps to improve the outcomes of patients treated within the trial itself. This is a cartoon used with permission published in JAMA in 2006 that is intended to show that in an adaptive trial, it is actually the data that accumulate within the trial itself that determine the direction the trial takes from the beginning, shown by the horizontal oval at the top, to however it turns out to terminate, shown by the boxes at the bottom. So it's actually the data that determine the direction the trial takes, and in ways that are much more complex than just simply early stopping for futility or early stopping for success. So when we talk about an adaptive clinical trial or making an adaptation, we're talking about making planned, well-defined changes in key clinical trial design parameters during trial execution and in response to data from the trial itself to achieve goals of validity, scientific efficiency, or safety for participants. By planned, we mean that all the possible adaptations will be defined a priori. By well-defined, I mean to imply that the criteria that we will use to determine when to make those changes are also defined a priori. We're talking about making changes in key clinical parameters like the number of treatment arms, the doses that are used, or the timing of interim analyses. We're not talking about the kind of minor changes like inclusion or exclusion criteria that are normally handled via routine amendments. And we want to do this in a way that we can achieve valid statistical inference, meaning we will understand what the effects of this flexibility will be on our chance of a false positive conclusion, on the power of the trial, or of qualitatively drawing the wrong conclusion regarding the effectiveness of the treatments that we're investigating. So during the design of an adaptive trial, one often gets a greater clarity regarding what you're trying to achieve than occurs during the traditional clinical trial design process. So for example, we need to be very clear. Are we trying to prove the concept of a treatment, meaning that it has potential efficacy, Are we trying to find the right dose, for example, in a phase 2 dose-finding study to carry forward into subsequent confirmatory testing? Or are we trying to confirm benefit of a specific dose or treatment algorithm that has been previously identified? I would suggest to you that a statistically significant p-value is never the goal of a clinical trial, because it is so easy to get a statistically significant p-value and still be left uncertain regarding whether the treatment ought to be used in practice or in what population, or whether the treatment or drug should be carried forward to the next phase of clinical development. We're going to take frequent looks at the data because the goal is to learn efficiently from the accumulating data within the trial itself. This contrasts the traditional approach in which you minimize the number of times you look at the data. We're going to be talking about trials that are adaptive by design, so everything is defined a priori. This will allow us to simulate these trials and understand their performance before we ever enroll our first patient. And we're going to use extensive simulations to adjust the characteristics of the trial design to balance our goals in the scientific development program against our resource limitations and other practical considerations. So this figure shows the general structure of an adaptive trial. We're going to begin our data collection with some initial allocation and sampling rules. For allocation, think of things like the randomization proportions, and sampling rules might be the number of patients we're going to enroll before we take a first look at the data. At that point, which we call the run-in period, there's nothing to learn from because we haven't gathered any data within the trial. We are then going to analyze those available data. Those data will be incomplete. There will be missing data. There will be patients who have been treated but who have not yet experienced their final outcome. And we're going to ask whether we've met one of the pre-specified stopping rules for the trial. Usually that doesn't happen early, so we can then use that partial data to revise the allocation and sampling rules to gain some sort of additional efficiency and then continue in our data collection. This process continues in a circular fashion until we meet one of the pre-specified stopping rules for the trial, one of which is always that we've met the maximum allowable sample size for the trial. Other stopping rules may be to stop if there is evidence of unequivocal benefit, or we've identified the right dose to carry forward into phase 3 testing, or alternatively evidence of harm or futility in continuing the trial. So why do we do this? Fundamentally, we go to this additional effort in designing more complex trials to avoid getting the wrong answer, meaning drawing a qualitatively incorrect conclusion, such as the conclusion that the trial does not demonstrate efficacy when in fact the treatment is efficacious and has a clinically important benefit. We also do this to avoid taking too long to draw the right conclusion. For example, taking too long in the time of the trial, the number of human subjects that are enrolled, or the resources that are used. It is a very unfortunate thing when we take too long to determine that treatment is ineffective and therefore squander resources, time, and put patients at risk without the potential for benefit. So one of the important advantages of an adaptive design can be the efficiency with which it fails when in fact the treatment has no clinically important benefit. When we're participating in the adaptive design process, one of the things we ask the clinicians and the scientific domain experts to do is to think about design decisions they might regret at the end of the trial. So if you think about clinical research in general, and specifically in the setting of neurologic emergencies or neurologic disease, there is a very large fraction of trials that, when conducted in the confirmatory space, meaning we're trying to confirm benefit that's already been thought to be demonstrated, we fail to demonstrate that benefit. So many confirmatory trials fail despite promising learn phase results. There have been 20 to 30 failed phase 3 trials of agents thought to be promising for modification of Alzheimer's disease, for example. Now, investigators can actually anticipate design decisions they would wish they could take over if a trial were known to have failed. So we ask people, assume you designed the trial the way you planned originally to design it, and at the end I tell you the result was suggestive of some effect, but in fact failed to meet the primary criteria for success. what design decisions do you wish you would take over? And those design decisions are very promising targets for adaptation, because if the trial was tending towards futility or towards an indeterminate result, those are the areas in which we might want the trial to do something different. Let me give you a concrete example. In a dose-finding study, very often people come to us thinking they know what the right dose is within a very narrow margin. For example, they think it's going to be a 1, 2, or a 4 milligram dose that will be most promising. And if I ask them, let's suppose that you run that trial and the 4 milligram dose has some evidence of benefit but fails to meet the criteria for success, what design decision do you wish you had taken over? And if they say, well, I wish I tried an 8 milligram dose, then the trial ought to automatically open up the 8 milligram dose. If the 4 milligram dose shows some benefit, a good safety profile, but not sufficient benefit to warrant clinical use. So you want that to happen automatically rather than having you be forced to wish it was a decision you'd been allowed to make, but you couldn't because you had narrowed the trial design and fixed it to consider only a maximum dose of 4 milligrams. Now, historically, most physicians have found that when they work with biostatisticians, they're told you can't do things this flexibly because they believe, the biostatisticians, that in order to achieve a reliable and valid conclusion, you need to have a relatively fixed or static clinical trial design. And this assumption has become a fundamental part of the culture of biostatistics and clinical trials methodology. The reason that exists is because in the 40s and 50s, when much of the current statistical mathematics was being developed, one had to make very rigid assumptions about the way that the experiment was conducted to allow the calculation of the risk of a false positive or a type 1 error. And now that we can model much, much more complicated experiments, the statistical culture and thought, frankly, has not caught up. So when is this approach most valuable? It's most valuable when you have some outcome of interest or a biomarker that you believe to be important, that it becomes available to you as an outcome for an individual patient in a relatively short period of time relative to the time it takes you to conduct the entire trial. So you have to be able to learn something from your early patients that allows you to improve the way the trial works for the later patients. It's also valuable when there's substantial morbidity, costs, or risks associated with the trial, so the additional design work is warranted. When there's large uncertainty regarding the relative efficacy or adverse event rates, so you don't know very much, so you have to be able to modify your design. It has to be logistically practical, and we'll spend a lot of time in design activities talking about how to do this. And you need to be able to secure the buy-in of stakeholders, which frankly has gotten much easier over time as there have been additional successes with this type of approach. There are counterexamples where you can't use adaptation, and the one I most commonly comment on is a prevention trial where you need to randomize all the patients to one or other prevention strategies and then wait a very, very long time to find out whether the disease of interest is actually prevented. Think of a cancer prevention trial. So why don't do this? I'm sorry, why should one not do this? One should not adapt if you can't figure out what the performance characteristics of the trial will be, but I hope to show you how you can do that. But figuring out the type 1 and type 2 error rates of an adaptive trial is more complicated. You generally need to do some custom programming or use statistical simulation software. And frankly, many statisticians graduating from PhD programs and now out in academia have never had any training experience or had the opportunity to do this kind of work. So this means for many academicians, when they approach their local statisticians or people they're used to collaborating with, what they will get is a lack of willingness to do this kind of work because it's harder and people don't like to try things that they've never tried before. There are also logistical issues. You need to be able to get your early data soon enough in order to inform the trial. You need to be able to randomize in a way that takes advantage of the information that you have acquired early on in the trial. And you need to be able to modify things like your drug supply. If I add an 8 milligram dose, I better be able to get the 8 milligram formulation out to the study sites in time, for example. So this table summarizes general differences between a traditional approach and the more flexible approach that characterizes an adaptive design. Generally, we make more use of more frequent interim analyses. We may use variable randomization ratios instead of a fixed 1-to-1 or 2-to-1. We like to consider more than 2 arms. That does 2 things. First of all, it increases the chance that you're going to include an arm that is actually effective. It also allows the one of the strategies we're going to talk about, response adaptive randomization, to make better use of the space you're giving it. We want to make complete use of partial data, so we may multiply impute missing data in order to use all the information available. And generally, these approaches are easier and more straightforward if we use an underlying Bayesian framework for the statistical approach, although that's not a requirement. Everything I'm talking about here is compatible with a frequentist approach, but is often more difficult to implement. And then again, we're going to need to use simulation of the clinical trial as we propose it, both during design to make sure that we've adjusted the parameters properly to get the performance that we want, and also to help communicate to people the performance of the trials that we are designing. Now, there are lots of different things we can do to modify the trial during its conduct with the potential to make it more efficient. The frequent interim analyses I've talked about. We can consider explicitly the relationships between early endpoints and late endpoints and learn about that relationship during the trial. That's longitudinal modeling. We can use response adaptive randomization to systematically shift the randomization proportions towards the arms that are most promising. This both improves the outcomes of patients within the trial and improves the precision of the treatment estimates we have on the arms that we're most interested in. And there's lots of other things that we can do. The general approach is to select a relatively small number of adaptations that give you the most benefit based on the particular characteristics of the situation that you're in. So there may be a situation in which an enrichment strategy is very important because only a subset of the population is likely to respond. It may be response adaptive randomization is very important, and there may be other situations where a different adaptive strategy gives you the greatest increase in efficiency or validity or ethical balance for your trial. And so the adaptive design process is one of identifying the best strategy for the particular experiment, testing and tweaking its performance, and verifying that you're getting performance that is appropriate for the scientific and clinical context. So when one selects various stopping rules and adaptive allocation rules from the long list of things that we can do, how do you figure out whether your design is any good? So you do that through simulation, and I'm going to show that a little bit later after I dive a little bit more deeply into response adaptive randomization. So when we revise the allocation rule to focus on a particular subset of arms, we generally, but not always, choose to focus on the arms that appear to be most beneficial based on preliminary data. So that is response adaptive randomization, and we may use this to improve the outcomes of patients within the trial, to improve the estimation precision for the arms we're most interested in, and we may do this to sequentially address different hypotheses. Our first part of the trial, we may be most interested in identifying the best dose, or a dose that has a particular rate of adverse events. And then later we may use it in a different way to focus on some different aspect of the trial. An extreme example of response adaptive randomization is arm dropping, where you allocate 0 probability to an arm being randomized going forward. That drops the arm from randomization. So that is a type of response adaptive randomization. So here's a concrete example. This is a dose-finding study, and during the burn-in period, we're considering 3 different doses of the active arm, a low, medium, or high dose. During a response adaptive randomization stage of the trial, in this case from 300 to 700 patients, we keep a fixed proportion of patients on the placebo or standard dose, or standard treatment. That's to help us adjust for secular trends if they exist. And initially, we start with patients on the highest dose and fewer patients on the low dose. And then over time, you may notice that we increase the proportion on the medium dose. At 700 patients, we may feel that we have sufficient information to say that of the doses being selected, the medium dose is the most promising, and at that point, we are doing a direct comparison of the standard or placebo treatment against that medium dose. And then at 1,100 patients, we may have enough information to say that the active arm with the medium dose is better than the standard or placebo treatment, and from then on allocate all patients to the treatment recommendation of the medium dose. This is an example of a trial that learns over time from an agnostic period during the burn-in to a learning about dose selection during the response adaptive randomization, to a confirmatory phase of stage 3, a phase 3 phase, if you like, or stage, and then to a treatment recommendation, which will be a form of clinical decision support. So this is a learning strategy that leverages response adaptive randomization. This has been addressed in brief in a article that Dr. Moyer at the University of Michigan wrote with myself and Don Barry, that simply points out that this addresses a common misconception among participants that the purpose of a clinical trial is to improve their outcomes. So many people enroll in clinical trials under a so-called therapeutic misconception that the purpose of the trial is to actually treat their disease. Response adaptive randomization allows us in part to actually help to achieve that goal of improving the outcomes of patients treated within the trial. So now let's come back to simulation. You can really only understand the performance of one of these trials if you can simulate it and simulate it under a wide variety of possible truths regarding how the patient population and the treatment behave. One does not know ahead of time, obviously, whether your treatment works or what the adverse event profile is, or necessarily even what the rate of patient enrollment will be. So a good adaptive clinical trial is a trial that does something reasonable and gets the right answer regardless of what the truth turns out to be. In order to figure this out, we need to stress test the design by simulating its performance under a wide variety of assumptions regarding possible truths. I want you to think about the way, for example, you might verify the design of an airplane. So in designing an airplane, you might want to verify that it flies very well in hot weather, cold weather, high altitude, low altitude, rain, when hit by lightning, etc. Some of those experiments we do in real life. Many of them are done on computers before we ever put a person in that airplane. This is exactly the same. We want to simulate the trial under a wide variety of possible truths to make sure it performs well before we ever put the first patient in the trial. It has been said that before you put the first patient in a trial, you should have subjected thousands of virtual patients to that trial to make sure it's a reasonable experiment. So this diagram shows the strategy for simulating an adaptive trial. So first, in the upper left, we're going to make assumptions regarding what the truth happens to be. We get to play God. And we make assumptions regarding population characteristics, accrual rates, how well the drug or treatment works, its safety profile. And under that set of assumptions, which we're going to allow to vary from simulation to simulation, we simulate thousands of clinical trials, and we ask if this particular truth was in fact the way nature worked, would the trial we've designed behave pretty well? We look at example trials to make sure it does something reasonable at each stage, and we look at operating characteristics, by which I mean summaries of the number of patients enrolled, the number of patients allocated to the different treatments, the outcomes of patients within the trials, and the rate of getting the correct answer, whether it's a positive correct answer or a negative correct answer, and then the various errors. A type 1 error when we falsely conclude a treatment works when it does not, and a type 2 answer, which is error which is related to power, which is the fraction of the time we fail to detect a true treatment effect that is clinically important. So I'm going to give you some examples of the results of simulation so you can understand how that will help you judge whether a design is a good design or not. But realize that early on in adaptive design exercise, when you simulate the trial, it's not going to work well. And then you get to play with the parameters and the decisions and the adaptations in order to iteratively create a trial design that performs well and is consistent with the logistical and resource utilizations that exist in all clinical trial settings. The goal here is to kill as few people as possible while we learn the truth. This is a cartoon borrowed from the New Yorker. It suggests that Mr. Dudson didn't do well and was allocated to the placebo group. There's an unstated assumption that being in the placebo group is bad. I will tell you that being in the placebo group is often a very good thing to happen to you because you get all the benefits of the extra attention from the clinical trialists without any risks of the experimental therapy. The point here is you don't know which treatment is going to be the right treatment, so you want to design a trial that does something reasonable and accurate regardless of what the truth turns out to be. So the example that I'm going to use is an actual clinical trial. It's currently enrolling patients, and it investigates an amino acid supplementation in patients with severe sepsis and septic shock. So we're enrolling adult patients with severe sepsis and septic shock. This is a phase 2 dose-finding study of L-Carnitine to improve end organ function and survival. We want to find the most promising dose of L-Carnitine, and we want to figure out whether or not the treatment effect of L-Carnitine is sufficiently promising that we ought to do a phase 3 trial. So the idea here is that we have a clarity of goal. It's not just to figure out if there's any signal here. It's to figure out, is there a dose that is sufficiently promising that we should invest the time and money and patient risk into a phase 3 trial. And as we do this, we would like to enroll more patients into the doses that are most promising, both to be beneficial to those patients potentially and to improve the efficiency of the trial. The background is that L-Carnitine is an important part of energy metabolism. It's in deficit in patients with severe sepsis and septic shock. That energy deficit is thought to be a common underlying feature that leads to multi-system organ failure, which is itself associated with death. So we're going to use something called the SOFA score, the Sequential Organ Failure Assessment Score, to measure the burden of multi-organ system failure in these patients. The baseline SOFA score when the patient presents is an important predictor of mortality. The hope is that an effective treatment that addresses this multi-organ system failure will be manifested by a change in the SOFA over 48 hours. So a good treatment would tend to lower the score in the first 40 hours, and ineffective treatment or in a patient who is trending towards death, the SOFA score would tend to go up in the first 48 hours. However, in order for the drug to be approved in a phase 3 clinical trial setting, we would have to show benefit in 28-day mortality. So we have two different outcomes we're interested in. The change in SOFA score, which is approximate endpoint, that may help be most informative in helping us select the right dose. But the decision about whether this drug is promising enough to test in a phase 3 trial really has to be dependent on 28-day mortality. So there's a question that's been posted from Dr. Levine. It says, is there a risk in adaptive design by focusing on more immediate and robust effects at the cost of missing information about potential longer-term beneficial effects at lower doses? So there are several pieces to that question. Any early data, because it's going to be sparse, is going to be inherently less informative and noisier. And the simulations help you understand any risks associated with moving your trial in a particular direction based on early data. However, we don't ignore the later data that accumulate. So for example, if a particular dose looks most promising early on and we put more patients on it, we will actually learn more quickly whether that early performance is a fluctuation because we're going to put more patients on that dose. And if it decreases over time, for example, due to regression to the mean, we will shift the randomization back. So the statistical modeling and the simulations help you understand the risks of adaptation and to mitigate those risks. And generally, that risk is lower than expected. The second piece implied by what I've seen you written has to do with early outcomes versus late outcomes. And this really comes back to this question of whether it's plausible or appropriate to use early outcomes to drive late outcomes. And what we generally do is never rely on early outcomes, but allow the data accumulating within the trial itself to help us understand the relationship of early to late outcomes, and then to only utilize early outcomes when we've demonstrated within this particular trial that they are highly predictive. And I'm going to give an example of that because we're going to use the relationship of SOFA score to 28-day mortality in this particular example. So the outcome measures in this example are the proximate change in SOFA score over 48 hours, negative is good, and survival at 28 days, and obviously a higher survival is good. We're doing dose finding with 4 arms, a 0-gram control, and 3 dosing arms, 6, 12, and 18 grams. You give a lot of this stuff, it's an amino acid. We have a maximum sample size of 250 patients, largely dictated by the resource limitations of the NIH funding mechanism. And we're going to start looking after 40 patients, so we have a run-in of 40 patients, and then we're going to look at every 12 patients. So this is the frequent interim analyses that we're talking about. The efficiency that we're hoping to gain is based on the efficiency of allowing the change in SOFA score, the thing we think to be most proximately affected by the treatment if it works, to guide the allocation of the dose. So we're going to randomize future subjects preferentially to the dose of L-Carnitine that appears to have the greatest benefit on SOFA score. And this will allow us most rapidly to focus on the most promising dose. But we're going to ask the question about whether this trial has a success or is futile based on the effect of that most promising dose on 28-day mortality, because the success or futility of a phase 2 trial really depends on whether or not you know whether you should do a phase 3 trial, and the phase 3 trial is only worthwhile if the selected dose of L-Carnitine has a sufficiently large effect, beneficial effect, on 28-day mortality, which would be the phase 3 registration endpoint. So we're going to simulate this trial. Now, I have not told you all of the futility rules and early success stopping rules that are included in this design. This has been published in Critical Care Medicine, and all the details are there. So I want to orient you to the simulation results. These are very crude simulation results, crude in the sense that there's a lot more detail I'm not showing you in the interest of time. But we're essentially going to consider 3 different possible realities. One reality, which is called the no effect or null reality or scenario, is the situation in which the drug does nothing. It doesn't affect the SOFA score at 48 hours, and it has no effect on mortality, which is expected to be 40% in this patient population. They're very sick. We're going to consider something I'm going to call mild effect, although in fact if this existed it would be a wonderful thing, which is a situation in which there's a dose-dependent improvement in SOFA score at 48 hours with increasing dose of L-Carnitine, and that is associated with a decrease in mortality. So this is the positive scenario where the drug works and the mortality is decreased based on that. And we're going to focus on those 2 columns. Obviously, I have no way of knowing if either of these are true, but the idea is to design a trial that does something reasonable regardless of what the truth turns out to be. If one knows anything about sepsis, I can tell you most drugs don't work, so the null effect is a more likely scenario than the mild effect if we were going to predict what's happening. The goal here is not to predict, but to design something that works well regardless of what the truth turns out to be. So how does this design work? Under the null hypothesis, where there's no effect at all of the drug on either the change in SOFA score or mortality, the chances that the trial falsely concludes that there's a beneficial effect and picks a dose is 4.3%. So that's our type 1 error risk, which is controlled at less than 5%, which is reasonable in a phase 2 trial. I will tell you that the traditional level would be a 5% two-tailed control, or 2.5%. So this is actually a little more than the normal alpha or type 1 error risk of 5%. This would really correspond to about an 8% two-tailed type 1 error risk. Under the scenario where the drug does nothing, there's a 43% chance, meaning 43% of the simulations stop early and don't enroll the full 250 patients. That saves patients, saves time, and allows the investigators to know the drug doesn't work and to move on to the next most promising treatment. The average number of patients that are required for the trial is about just under 200. And when it makes a mistake and selects a dose, it's sort of random what it selects because it's responding to noise. So about a third of the time it picks the highest dose. So on average, this trial may stop a year early, for example, because the design appropriately figures out there's nothing here worth investigating. Under the mild treatment effect, which obviously would be great, there's about a 68% chance that it stops early for success, allowing you to move forward with the design and implementation of a phase 3 trial, and almost never, less about 0.1% of the time, stops early for futility. The power of the design to detect this treatment effect is over 90%, and 99% of the time it correctly picks the 18-gram dose as being the correct dose to carry forward. I can tell you that many traditional approaches to phase 2 dose-finding trials are actually very, very unreliable in selecting the correct dose, and the approach we're using here is substantially better. So I urge you, if you're ever looking at a dose-finding trial, don't look at the power just in terms of finding out whether there's a treatment effect. Make sure you know the probability that the trial will find the right dose, because it is thought that many subsequent phase 3 failures are related to bringing the wrong dose forward from phase 2 into phase 3. Now, how about the treatment of patients within the trial? Under the null scenario, where it doesn't matter what you get because nothing works or harms you, about a third of the patients get the control treatment, and the rest of the treatments are about evenly distributed, which is fine because none of it matters. Now, for the mild effect strategy, in which it's much better to get the highest dose, about a little less than a third of the patients get the control treatment because we need that for the validity and the reliability of statistical inference. But notice how the allocation of treatments is strongly skewed towards the highest dose. This is because the trial utilizes response-adaptive randomization and therefore was able to learn relatively early that the patients really ought to be allocated to the highest dose if they were allocated to the active treatment arms. This not only improves the outcomes of patients within the trial, it improves the precision of the treatment estimate for the dose you're most interested in, which is the 18-gram dose, and essentially gives you the same power, because you've got half the patients on that dose, as if you somehow magically knew that was the right dose and just did 50-50 randomization between that dose and a 0-gram control allocation. There's been a question posted about whether there are special ways to handle adaptive designs for diagnostic strategy trials. There are, and many of the general concepts I'm discussing today can be modified for diagnostic strategy trials, especially the futility and early stopping rules, and we can certainly talk about that in a different context. I'm focusing on therapy trials for the purposes of this discussion, but for those of you who are participating in the course, we will talk about a variety of different kinds of trials, and we will apply these same concepts to diagnostic strategy trials if that's one of the projects that's brought forward. Okay. So again, another way of looking at the allocation, this shows you under the hypothesized mild effect scenario that most of the patients who get an active drug get allocated to the highest dose. It shows you the variability from simulated trial to simulated trial. This helps you understand whether or not it's a good design. This slide shows you what fraction of the time it picks the 18-gram dose. That's the big sort of olive bar to the right. It also tells you the accuracy of the design in estimating the true treatment effect. Again, we use these kinds of diagnostic plots after simulation to understand whether the design is a good design or not. I am showing you the performance of a design that was ultimately submitted for funded, has been funded, and is being run. What I'm not showing you is all the false starts where we looked at these and said, wow, that's not a good design, we need to fix that. This trial is currently being run. It's been funded by the National Institutes of Health, the National Institute of General Medical Sciences. The principal investigator is Alan Jones, who's the chair of emergency medicine at the University of Mississippi. It is currently enrolling patients, and I think I can say, without disclosing any information, that it's currently in the stage where it's using response adaptive randomization to more efficiently allocate patients between the active treatment arms, meaning they've gotten through the run-in period. So this is going very well. Okay. So now I'm going to shift gears a little bit, and we're going to have time for questions at the end and talk a little bit about the logistical or administrative structure required to do something like this. In order to have the trial learn from early data, you have to be able to get a subset of the data that informs the adaptive algorithm gathered soon enough so that you can learn from it. So here we show a possible organization of a multi-center adaptive trial where we have a number of clinical trial sites, and those have to be set up so that the basic outcome data, so for example, for the sepsis trial, you'd have to know the patient, basically their SOFA score at 0, 48 hours, and when we get to 28 days, whether or not they lived or died. You'd have to have that subset of the data available relatively quickly to the data management or contract research organization who's been doing data analysis. You don't have to have the full data set. You just have to have everything necessary to inform the adaptive algorithm. It is understood that those data will be not verified or monitored. There may be errors in those data, and as long as those errors get corrected over time, this trial gets smarter and smarter over time. Those data elements are fed up to a data analysis function that runs the adaptive algorithm, which will determine, for example, whether or not any of the pre-specified stopping rules have been met and what the new randomization proportions are. That needs to be fed back to the randomization system. It may also change your drug supply needs so that the next patient who comes in at any of the sites has a randomization proportion that is informed by that adaptive algorithm. And so, for example, in the trial I just described, that is being updated once a month. The adaptive design should be overseen by an independent data safety monitoring board. The DSMB does not make decisions about any of the individual adaptations, but is there to verify that the trial is being conducted as intended and that the design remains appropriate. This is the usual function of the data safety monitoring board. And again, you may have a steering committee and a separate sponsor depending on the context for the trial. Often people ask questions about the IRB review of these trials. So the first comment I would make is that IRBs are generally very favorably disposed towards adaptive trials because in general the adaptations are selected to improve the ethical balance and decrease the avoidable risk for research subjects. Sometimes questions are raised regarding the degree with which a research subject can understand the complexity of this trial. And if we ignore for a second the fact that research subjects often don't understand the simple trials that we're in, we have found it very effective to simply say that information gathered during the trial early may be used to alter the trial during its conduct to improve the efficiency or safety of the trial. We certainly don't give any specifics regarding the adaptive algorithms. Because the adaptations are defined a priori, the implementation of an adaptation, including dropping an arm, is not a change in the trial protocol. So there's no new review by the IRB when the adaptations occur because the IRB has to review and approve the entire study plan. Now, they may request to be informed when something notable happens, and you can certainly do that when appropriate, but it needs to be very clear that the IRB needs to sign off on the entire plan because that is the pre-specified protocol for the trial. Now, amendments are different. These unplanned things that happen during a trial in which you change inclusion-exclusion criteria and those sorts of things, add or drop sites, those are not pre-planned. The IRB has not had an opportunity to approve them, and therefore those do require separate IRB approval, just like they would for a non-adaptive trial. So the irony here is that little changes, sometimes minor amendments, require an IRB review, whereas big changes, the adaptations, because they are pre-specified, are reviewed and approved only once. I talked a little bit about data safety monitoring boards. The only difference in the constitution and behavior of a DSMB in this setting is that they need to have the sufficient expertise so that they can understand and verify that the adaptive design is being implemented as planned and that the design remains scientifically and ethically appropriate. So you may need to include personnel you might not include in a normal DSMB. But it's really the trial that's adaptive. It's not the DSMB, and therefore it's also equally important the DSMB understands that their role is not to redesign the trial to create a different design, because doing so would invalidate all of the work that was done up front to understand the appropriateness and the operating characteristics of the pre-specified design. There's another question we often get asked, has to do with the regulatory status of these designs. This simply shows you two of the guidances that are related to this. The FDA has a guidance for the use of Bayesian statistics in medical device trials. That's to the left. It includes a number of aspects related to adaptive design. These approaches are extremely well liked, appreciated, and supported by the device branch of the FDA, which is a very progressive and flexible group. The guidance document to the right is from the biologics and the drug branch, the drug obviously being the largest branch. And it sets out standards for adaptive clinical trials and confirmatory trials. There is no doubt that adaptive approaches are well appreciated and have tremendous advantages in learn phase, so phase 1 and phase 2 trials. That's never been a regulatory issue as best I can tell. So this guidance focuses on the considerations when you want to use an adaptive design to show unequivocal confirmatory evidence of a benefit for regulatory and licensing purposes. It's a very well-written guidance, and I can tell you that in recent interactions with the FDA, we found them to be extremely open-minded and supportive of these approaches when the approaches can be demonstrated to have good operating characteristics and to have advantages for the patients enrolled in the trial. So, in conclusion, not all trials need or should have adaptive designs because this entails additional design work, it requires additional expertise, and it entails some additional logistical and implementation considerations. But when used appropriately and in the right setting, these kinds of approaches can improve the efficiency and reduce costs of trials. They can maximize the information obtained because, for example, you put a greater fraction of your patients on the treatment you ultimately are most interested in. And they can minimize risk to the investigator, the subjects, or the person or entity sponsoring the trial. Your decisions regarding the design of the clinical trial should be based on objective evidence that the selected design is better than another design, not based on habits, such as the hot habit of assuming that any design decision made before you knew anything need to be held constant throughout the trial. An adaptive design will not save a poorly planned trial or an ineffective therapy, although it may help you be more efficient in identifying ineffective therapies, identifying futility, and then reducing unnecessary risks and expenses. So with that, I'd like to stop and open it up for questions. You can type questions and I'll do my best to address them. Thank you very much. And perhaps I can start by asking Dr. Levine and Newman-Toker whether or not I addressed the questions that they previously entered. I'm a little concerned. I have a little message that simply says multiple people are typing. So Dr. Levine asks whether adaptive design is primarily used in phase 2. So adaptive designs of various types are used in all phases. very, very often in phase 1. I think it's almost a non-issue in phase 1. In phase 2, they're very, very frequently used. And you must remember that from a regulatory point of view, if there's an increased risk of a false positive result in phase 2, the FDA is not very concerned about it because they're going to make you prove the drug or device works in phase 3 or a definitive or pivotal trial. So the advantages of an adaptive design are greatest when there's uncertainty regarding how the drug or device should be used. There's obviously greater uncertainty in phase 2 than phase 3. So I think the use of adaptive designs in phase 2 is very well accepted and often highly advantageous. Now, in phase 3 trials, there's a tremendous premium on making sure that you understand exactly the risk of a false positive result, because from a regulatory point of view, people are most concerned about the possibility of approving a drug or device that's truly not effective. And in this area, there's been a lot of recent work and movement in the regulatory agencies regarding how to do this for an adaptive design, but I think that's an area, a growth area that's at an earlier stage than the phase 2 work. A question has been posed, says, I'm curious about statistical methods to analyze the final data when the design is adaptive and randomization allocation shifts over time. That's a great question. So one can analyze the end data. This is going to seem like a funny thing to say, almost any way you want. And the reason I say that is because when you simulate the trial, you're going to include in the simulations whatever approach you're going to use to the final analysis. So if you're going to do a multivariable logistic regression with 5 covariates at the very end, you need to simulate that and then count up how often that gives you the right answer or the wrong answer under the different scenarios to show that you have the precision and the operating characteristics that you want. It is very common for us to design adaptive trials that use a Bayesian machinery to run the trial, and then at the very end of the trial, the final analysis is a traditional frequentist analysis. It's not my preference, but we do that very frequently. It often makes people more comfortable with the final analysis. But the overriding issue is that you need to verify through simulation that that analysis that you intend at the end of the trial has the correct error rates and good precision, even when applied at the end of an adaptive design. Now, there are kinds of adaptive designs where it's very well worked out how to do this. For example, in a group sequential design, it's very well known how to correct the alpha cutoff at the last analysis to control type 1 error risk. But for a design in which you're shifting randomization proportions over time, it's less well worked out, and generally you need simulation. Other questions?
If there are no additional questions, I'd just like to thank everyone for joining us today, and especially thank our speaker. Thank you very much, Dr. Lewis, for a great talk. If anyone is interested in CMEs, please use the link again to the evaluation form below. And our next webinar will be on May 21st for the Foundations track, and the topic will be about information you need to start writing your protocol. So thank you again to everyone. Thank you, Dr. Lewis.